Hi guys, welcome to the Diatom annotation tutorial for the microbe directory. Bear with me, it's going to jump around a little bit. Everyone has their own annotation process. So first I usually start by, I make sure I have a notebook or paper where I have all of the characteristics I need to look for written down so that I write them down as I go along, and I write down which sources I got what information from just so I know where I'm getting my information from. And I keep that next to me, I write it all down, and then at the end when I'm finished I put everything into the Kobo form, and I usually have the form just tucked away, ready to go, minimized. So I usually start off by just Googling my assigned species and opening all of the reliable looking sources that come up, and I just open them all at once and I start annotating what I find. So this is the first source we have. It's the Diatomsorg source. And I just start looking at what information I can get from it from the list that I need to get. So reading all this and looking at this information and reading it, I've already got microbiome, which is freshwater. I can annotate lakes and rivers. I can do brackish and marine coastal waters. I also have that its growth is colonial from this little table, as well as the opening sentence. I have that its lifestyle is benthic, epipelic, but it can also be planktonic. From the pictures and from just the information in this page, I know that its morphology is fusiform based off of the definitions and the diagrams that we have on Coda, which I always keep open when I'm annotating just so I have access to it as soon as I need it. Its symmetry is pennant, and so far that seems like everything I was able to get from this source, so I moved on to the next one. The next one is more so just taxonomy, so I usually keep that tucked away in case I hit a wall and I need to look up synonyms to see if there's maybe information useful there. So moving on. Once this source showed me that it reproduces both sexually and asexually. It also verified the colonies as well as habitat and lifestyle. So benthic, but also displays planktonic. That seems like everything we got from this one. It also talked about morphology, so I didn't know that word, so I looked it up. And based off of the definition of that word, I know that it correlates with how we define fusiform. So I'm confident in that annotation. So far, I take a look at what I have and what's left. I have microbiome, growth, lifestyle, morphology, symmetry, mobility, type of reproduction, and so far that's all I have. I noticed from this source that there seems to be maybe a salinity level that's not normal, as you can see here and here. So I know that I'm gonna keep in mind that this might be a halophilic species, and I'm gonna look into that later on. I also like to open all of the useful links that are relevant to my species, and then just like searching the species over and over in each of the sources. Some of them were useful, some of them were not, and I kept the ones that were useful open. Next, I start specifically Googling the aspects that I'm looking for. So here I added cell wall. And I found this article from TNDEF Online, this study, which from it I saw that there were no secondary structures, there were no bristles, there were no spines, so I annotated no for secondary structures. Now, pigmentation is, as of right now, is a little tricky, just because that's not something that's usually going to be listed for you when you're looking for a specific species, but it's important to know that there are certain pigments that are always going to be found, such as chlorophyll A, C, fucoxanthin, beta-carotene, and a few others. So there's this source that kind of just lists all of them and tells you which ones are more common, or which ones are standard, whilst others may or may not be found. So we're seeing that these are needed for survival, so I usually just always annotate them as present, whereas these, there seems to be variability with them, so unless I find a source specifically saying that my diatom produces them, I will not annotate that it does. I did not find any sources about my diatom producing these pigments, so I just annotated. So I'm just going to annotate for these. So we have pigmentation. Next is biofilm. I annotated yes because of the fact that my species is a marine benthic diatom, and those are known to produce large amount of EPSs, which are used in their biofilm, which helps with attachment. And there I found two general sources discussing how marine benthic diatoms produce these exopolymeric substances in order to aid in their attachment. I also use this to help me with the annotation for content production, because I'm seeing that there are carbohydrates and proteins that are being produced through the production of EPSs, so I'm going to annotate yes for content production and annotate carbohydrates and proteins. So next I saw that through two sources, I saw that there seems to be some halophility with this species, so I just Googled the species and the word halophile to see what comes up. I found on ResearchGate.net there seems to be a few sources talking about how this species is salt tolerant, so I wanted to see if there were any other sources stating that. I found this one, but it wasn't really available to me, so I Googled the title to see if there was anywhere else I can have access to it, and I got Semantic Scholar. So using this source, I'm going to annotate yes for extreme affinity, and I'm going to say it's a halophile. So now everything I have left is spores, symbiosis, pathogenicity, antagonistic activity, resistance to UV radiation, toxins, and metabolism. I usually just make a note of everything I have and everything I don't while I'm doing my research. So I just keep Googling my species with whatever characteristic I'm looking for. With spores, I didn't find anything that was verifiable or anything that actually stated that it was sporulating, so I'm going to annotate unknown. Same for pathogenicity, because I found one source saying that it doesn't have any harmful effects on record, but then another source I found states that it could be a harmful species, and then a third one about it having harmful blooms. So for both pathogenicity and toxicity, I put unknown because from the research I found, I have no idea what other organisms it affects, if it does any in a negative way, or if it produces toxins of any kind. There seems to be some ambiguity with that, so I'm annotating unknown for both. And that's just me checking everything off to make sure I'm covering everything. There's no symbiosis with this species. It was always recorded to be as attached to a sort of substratum such as rock or just free floating. And there was no record of it having a symbiotic relationship. So I'm going to annotate no for symbioses. Some of these things you won't be told straight to your face in an article, like no article that's researching a specific species, or not all of them, are going to say, so this species has no symbiosis, it has no pathogenicity. You just have to see what's coming up, what information is coming up about your species, and annotate based off of that. With antagonistic activity, I did not find any sources stating that there was antiviral, antifungal, or antibacterial activity. That doesn't mean there isn't any, because that's something that usually gets less focus, whereas something like symbiosis is more often mentioned because it's about how that species lives. So with antagonistic activity, because of the fact that it wasn't stated anywhere, I just put unknown because I have no idea if it does or does not have antagonistic activity. There's no research for it. And resistant to UV, same thing. I put unknown because none of the sources I found ever mentioned it. I specifically would search UV, UV resistance, UVA, UVB, and see what sources come up, if any, and nothing. So for metabolism, I tried several different search parameters. I tried energy source, I tried metabolism. Sometimes I put just the word I'm looking for, like I try heterotroph to see if it comes up in an article, if they come up together. That didn't work out. I tried it with chemotroph, phototroph, every combination I tried didn't seem to work. So I annotate unknown because I can't find a source that is explicitly stating its metabolism. So overall, I spent about 40-50 minutes on this annotation. Some take longer than others. Once you hit half an hour, and you could keep looking, and if you're seeing that you're not really finding the information you need, then, and that the sources are getting like a little sketchier, less reliable, and there seems to be like a lot of repetition where you're coming across the same sources over and over again, then it's safe to assume that the information you're looking for has not yet been published. So I have all this information written down, so I'm going to go ahead and open the Kobo Form and fill it in. So I'm going to copy and paste my scientific name. Diatom is a eukarya, algae, microalgae. It is a diatom. So water. I found sources saying brackish, estuaries, fresh and marine, lake and river. I did not find a specific marine biome, so I'm going to click unknown. For growth, it was colonial. There was both of these. The morphology was fusiform, symmetry was pennate, secondary structures, it did not have any. It does produce pigmentation. I'm going to annotate the reliable, ever-present pigments. I did not find any sources specifically stating the others. Mobility I have as motile. I did not find a mobility structure. Well, it was the RAF. These are more so for microalgae. So I'm actually going to annotate NA. I'm going to annotate yes. Is it an extremophile? Yes. Halophile. It had both sexual and asexual reproduction. Does it form spores? I did not find this information. Content production was a yes. And we found, at the very least, carbohydrates and proteins. Resistant to UV, unknown. Toxins unknown, and metabolism, I did not find it. ZBib, I usually open it in an incognito window because otherwise it starts to lag, but every time I use an incognito window, I get my citations pretty quick. And then it's just a matter of copying and pasting all of these sources into ZBib and pasting them here. Any notes or comments that you think are necessary, and I usually save draft, but you could submit if you want. And that is the diatom annotation. Hope it helps.